Gli inizi sono potenti. Tanto è cominciato qui, nella città del vento e della scienza. Nella città ai confini, dove le barriere, se esistono, vengono soffiate via. Qui c'è un'energia unica che cambia il mondo, genera intesi, supera i limiti. Qui sono nate aziende che hanno colto idee e opportunità. Qui le menti brillano, pensano più forti, creano con una visione chiara. Qui, dove l'innovazione è al servizio dell'umanità, il futuro avrà una casa. Qui, dove inizia il futuro. Gli inizi sono potenti. Tanto è cominciato qui, nella città del vento e della scienza. Nella città ai confini, dove le barriere, se esistono, vengono soffiate via. Qui c'è un'energia unica che cambia il mondo, genera intesi, supera i limiti. Qui sono nate aziende che hanno colto idee e opportunità. Qui le menti brillano, pensano più forti, creano con una visione chiara. Qui, dove l'innovazione è al servizio dell'umanità, il futuro avrà una casa. Qui, dove inizia il futuro.
Buongiorno, sono Marco. Vi informiamo che potete seguire l'appuntamento in diverse lingue grazie a un servizio di supporto alla comprensione simultanea realizzato con intelligenza artificiale, accessibile direttamente dal vostro cellulare. Vi chiedo qualche secondo per spiegarvi come funziona. Per prima cosa, prendete il vostro telefonino e inquadrate il codice QR che vedete nel video. In questo modo potrete scaricare sul vostro cellulare l'app di Interprefy con cui viene gestita la traduzione. Vi lascio ancora un secondo per inquadrare il codice QR. Dopo aver scansionato il codice QR, si aprirà la app. Scrivete nel riquadro al centro la password che vedete qui e fate tap sul bottone con la scritta Connect in basso. Si apre così la vostra schermata operativa. Facendo tap in basso a sinistra, sulla scritta English indicata dal riquadro, si aprirà una schermata che vi permette di scegliere l'audio della lingua di preferenza, inglese o italiano. Usate le vostre cuffie e ascoltate o la lingua originale o la versione tradotta dall'intelligenza artificiale. Facendo tap in basso a destra, sulla scritta CC indicata dal riquadro, invece potrete scegliere di attivare i sottotitoli e leggere la trascrizione della lingua originale o della lingua tradotta con l'intelligenza artificiale. Per uscire dalla app sarà sufficiente far tap sul simbolo in alto a sinistra indicato dal riquadro e poi cliccare su Leave. Vi faccio vedere ancora una volta il codice QR per accedere alla app. La traduzione con intelligenza artificiale è in continuo sviluppo. Vi invitiamo a usare questo servizio per contribuire a migliorarlo. Grazie.
Hi, I'm Sophia, and once again I have the pleasure of guiding you through this year's festival. The theme of the 14th edition is Life Within, Conversations Across Sciences and Technologies. 3 days to explore how science and technology are reshaping life itself, how STEM disciplines engage with the humanities, and the new questions these encounters raise about our future. Precision medicine, artificial intelligence, environmental sustainability, robotics, biotechnology, and quantum technologies are just some of the fields we will be diving into. This is the thread running through Trieste Next, a program featuring more than 100 events in English and Italian, with over 300 distinguished speakers from around the world. The festival also includes the Science Book of the Year Award, over 70 activities for schools, and 40 open spaces and labs in Piazza Unita. Before welcoming the guests of the upcoming event, on behalf of the promoters and co-promoters of the initiative, I would like to thank all the scientific institutions of Trieste City of Knowledge and the institutions that have collaborated with us. The main partner of the festival, the partners, and the content partners of the event for their essential contribution. Thanks also to the media partners and technical partners. Finally, a special thanks to the 200 volunteers of this edition. Please also remember to check the updated program at www.triestenext.it or simply scan the QR code you see here. Enjoy the festival!
Buonasera a tutte e tutti, buon sabato sera, buon Trieste Next. Vi do il benvenuto a questa seconda serata speciale promossa dall'Università degli Studi di Trieste con il supporto della Regione Autonoma Friuli Venezia Giulia, durante la quale incontreremo un ospite d'eccezione, un premio Nobel per la chimica che ha vinto appunto nel 2012 assieme a Robert Lefkowitz e il professor Robert Kobilka. Prima di iniziare, chiamerei qui sul palco, come abbiamo fatto anche ieri sera, l'assessore Maurizio De Blasio, che invito. Buonasera, prego mi raggiunga. Eccoci qua, buonasera.
Grazie Marconi, sono sempre io, lo dico per chi c'era anche ieri e che quindi si sentirà il saluto anche oggi, saluto invece chi ieri non c'era. Immagino che tanti invece abbiano colto l'opportunità di assistere a questi due eventi straordinari. Ieri mi sono soffermato sulla divulgazione e sulla conoscenza. Oggi invece credo sia importante sottolineare l'importanza della ricerca, dello studio, e quindi sono lieto e onorato di poter presentare un incontro con un premio Nobel come il professor Kobilka. Ricerca che è indispensabile per tutti noi, per la crescita, per il progresso non solo tecnologico, ma anche civile, anche sociale. Quindi è compito di ognuno di noi come si può in qualche modo sostenerla e fare in modo che proceda. È chiaro che il primo compito è quello degli amministratori, è quello dei politici, ma è compito poi che spetta veramente al singolo cittadino. Quindi come ieri sera vi auguro una buona serata, un buon ascolto e un buon Trieste Next. Grazie.
Grazie a Maurizio De Blasio. Tra l'altro mi devo correggere, l'emozione di vedere questa sala piena. Ho detto Robert Kobilka, ma è Brian Kobilka, ovviamente ho sbagliato il nome dell'ospite principale di questa serata. Dicevo appunto Trieste Next, come sapete, è promosso dal Comune di Trieste, Università di Trieste, il piccolo gruppo NEM, Area Science Park, SIS, OGS, con la copromozione della Regione Autonoma Friuli Venezia Giulia. Questa serata promossa dall'Università, quindi chiamo a raggiungermi sul palco il prorettore alla ricerca nonché professore di chimica Paolo Fornasiero. Benvenuto a Trieste Next.
Thank you. Good evening to everyone. It's really a great pleasure and an honor to welcome all of you on behalf of the University of Trieste to this special event. We are hosting a great scientist, Professor Brian Kobilka from Stanford University, who received the Nobel Prize in 2012 for his groundbreaking research on G protein couple receptors and for the implication on human health. As a chemist, I feel really the privilege to address a few words tonight, and I want to be very brief because I know you're here to listen, Professor Kobilka. But let me start thanking Professor Kobilka to be here. I think this is an important event, extremely important event. Yesterday, here in this same room, we heard that science is under attack. And I think today we have the opportunity to dissipate some fog that is present in many scientific debates in this period. The opportunity to listen from a Nobel laureate, the discovery he made is a unique opportunity. And in my little experience with Nobel laureate, I have to admit they have always found great scientists, but also a person with a special attitude to interact with people. And this is also very important because public engagement today is a must. And his presence today here is a sign that Trieste Next is really on the right direction. The second thought I have is that the view of this beautiful theater full of people anxious to learn more about chemistry, biology, the language of the cell is really a positive aspect. I'm really happy to see many of you here. Last but not least important message I want to underline tonight is the role of young early stage, early career researcher in the development of science. Many of early career scientists are here involved in Trieste Next. And it's my personal, but I think also shared by many others, view that science proceeds by step and collaborative work is so important to tackle today's huge problems on energy, medical aspects, and vaccines, for instance, and so on and so forth. In this view, I would like to spend the last words on Marta Gianelli, a young and brilliant, motivated researcher in chemistry at our university. She just passed away 2 weeks ago in a tragic car accident. And our community, our university, our city was really shocked by this tragic event. Therefore, I ask you please to rise and have a moment of silence in her memory. Thank you very much. And now I do believe it's time really to listen Professor Brian Kobilka, who is going to talk about the language of the cell and his discovery. I want just to mention one thing. We have here the artificial intelligence that help us with translation. This makes our life much easier. Is a brilliant change in our life. However, understanding the language of the cell is much more complex, and as a chemist, I do hope to see the role, the importance of chemistry also in this very delicate process with important implication for the quality of our life. Thank you.
Thank you, Professor Fornasiero. And now let's welcome Professor Brian Kobilka, Nobel laureate for Chemistry 2012. Welcome to Trieste Next. Enjoy. Thank you very much.
So I want to thank the organizers for this invitation and the opportunity to speak to you tonight. I understand that the audience has a fairly broad interest, so I'm going to try to approach this from my perspective in terms of learning about the field myself and becoming a researcher in the field. So I'm going to first talk a little bit about my career to becoming a physician scientist. So that's a physician who also does research. As I'll mention later, I have an MD degree, but I don't have a PhD degree. And it's somewhat unusual these days for people with MD degrees to become very involved in research because they're so valuable in terms of patient care. Then I'm going to talk about my field, G protein coupled receptors. So we call them GPCRs. What are they and why should we care about them? And then a brief history of the field and some of my contributions. So first of all, I grew up in a small town in Minnesota. The town is called Little Falls, Minnesota. It was on the Mississippi River. So here's a aerial view of my hometown, roughly 7,500 population. And this is the Mississippi River. My father owned and operated a bakery called the Sanitary Bakery. And my first job was slicing bread. You can see how big the town is. So our home was the north end of town. I was born at the south end of town in this hospital. So it's roughly 2 miles or maybe 3 kilometers. As growing up, I was particularly interested in becoming a physician because 3 of my friends' fathers were the physicians in town. And I admired that they could help people out. They lived in nice houses. They lived on the river. So it was very attractive to me. And so I became interested in science because I knew I would need some biochemistry and chemistry and biology. And after graduating from high school, I decided to go to the University of Minnesota in Duluth, which is one of the coldest places in Minnesota. And it's right at the end of Lake Superior. Now, one of the first things that happened to me as I started my classes is I met my wife-to-be. I met Tungsan Kobilka, at that time Tungsan Tien, in biology lab in the first week. And we became friends, and obviously it evolved beyond that. We were married a number of years later on. Now, another important person in my life there was one of my early professors, Conrad Furling. And University of Minnesota Duluth was not a big research institution, and the professors didn't have access to graduate students, so they often had to rely on undergraduates to help them with research. And he really got me interested in trying some basic research in his lab. So what he worked on was an organism that was important in developmental biology at the time. They were called Chronomus tentans. They were these red worms. And they had what we called salivary glands that had polytene chromosomes. So they're very large chromosomes, and you could look at changes in gene expression in a simple microscope. So this was my first exposure to research, but it didn't convince me that I wanted to go into research, and in fact, I still wanted very much to become a physician. So going from Duluth to Yale University in 1977, and another big event in my life is that a year later, Tang-san and I were married in June of 1978. As a medical student at Yale, we were required an MD thesis, and nothing like a PhD thesis, but a simple question and addressed with simple mechanisms. And I chose to work on the diversity of genetic diversity of rotaviruses. So rotaviruses are one of the major causes of diarrheal illnesses in children. And I simply looked at the wide variety of genetic material in these different organisms. But again, it was interesting, it was very descriptive. I thought that at some point I might want to try a more challenging research project, but I knew I wanted to go on and complete my medical training. So after medical school in Yale, and in the United States, we do a residency, and I did a residency in internal medicine at Barnes Hospital in St. Louis from 1981 to 1984. During this time, both my children were born, my son in 1981, my daughter in 1983. And during my residency, I was particularly interested in intensive care. In a way, I want to be careful with the way I say this. It's like doing experiments. You have data coming at you very quickly. Patients are instrumented. You get a lot of information. You get blood tests back very rapidly. You make decisions about whether they're getting better or not, and you can change your therapy. And so I really enjoyed this. And a lot of the medicines that I was giving patients in the intensive care unit worked on a family of receptors called G protein coupled receptors. I knew very little about them at that time, but I was particularly interested in receptors for adrenaline. Nevertheless, because I was interested in intensive care medicine, I chose to study cardiology, to become a cardiologist. And I ultimately was accepted into the program at Duke, which at the time was one of the more interesting programs. It was a very large program, and they allowed their fellows to do a longitudinal or a longer research experience. So I started in Duke, and I had the opportunity to do research before my clinical rotations. And one of the reasons that I wanted to start in the lab was that I was tired of being on call. I wanted to spend more time with my young children, and I thought this research opportunity would give me a chance to do that. So I ended up choosing the lab of Robert Lefkowitz because his lab was working on receptors for adrenaline, or adrenergic receptors. I didn't know much more beyond that, but I thought, well, at least there's some clinical connection. Now, in retrospect, this might not have been a good idea because he had a huge lab. He had roughly 25 postdoctoral fellows and 2 graduate students. And I was the token MD student. He knew that I had almost no experience and would probably not succeed very quickly, if at all. But he himself has only an MD, so he would always take one MD fellow in his lab. So here's a photo of my family at the time. We lived in a very nice neighborhood, and there was a pond behind our house, and my 2 children at the time. So I ended up starting in the lab, and as I'll describe to you in a little bit, the kinds of work that I did, and I ended up, I planned to spend a year and a half in the lab. I ended up putting off my clinical rotations. I spent 2 years, 3 years, 4 years, and I really didn't become a cardiologist. I really just became a scientist. But nevertheless, I was able to fortunately get a job. And you'll understand why in a while. We were quite successful in some of the projects in the Lefkowitz lab. And at the very end of '89 or early 1990, we moved to Stanford, where I was initially a primary in medicine. That means my major appointment was in internal medicine. I had a joint appointment in a brand new department there called molecular and cellular physiology. I was allowed more than 75% of my time to just be devoted to research and patient care. So I attended on the medical service 1 to 2 months a year, and I did a lot of moonlighting. So I worked in emergency rooms to try to make extra money to support the family. My wife, Tung Sun, started medical school at this time, and we had a fairly big mortgage. So I needed to be able to earn extra money, and I still was sort of very clinically active in doing this. So anyway, that's sort of my path from growing up in a small town to ending up at Stanford with a fairly nice position in terms of having opportunities for both clinical and research. Now, when I joined the Lefkowitz lab, I mentioned that the field of G-protein coupled receptors was really quite new, and I was fortunate to sort of get in on the ground floor. But I want to tell you a little bit about what these receptors are and why they're interesting first, and then explain a little bit about the history of the field. So G-protein coupled receptors mediate cellular responses to the majority of hormones and neurotransmitters. So this is the basic mechanism that we understood at the time. We knew these receptors were plasma membrane receptors, that proteins reside in the plasma membrane of a cell, that when activated by a hormone or neurotransmitter, they could then engage proteins inside the cell called heterotrimeric G proteins. So why they're called G-protein coupled receptors, consisting of alpha, beta, and a gamma subunit. When activated by a receptor, the alpha subunit would release GDP and bind GTP, and then split into the alpha and beta gamma subunits. And these would go on to interact with other proteins in the cell. They could be enzymes or ion channels, and change the behavior of those proteins until they hydrolyze GTP, and then the alpha and beta gamma subunits would join again to form the trimer, and the whole cycle could begin again. So again, these are really important, particularly for cellular responses. So the majority of your body's responses to neurotransmitters and hormones work through G-protein coupled receptors. So what is a neurotransmitter? Well, these are molecules that are released from one neuron to activate a neighboring neuron. And they include things like adrenaline, dopamine, serotonin, you might be familiar with. And then hormones are small molecules that work at a distance. They can either be released and activate other cells locally, such as histamine, so your response to an allergic reaction. And then they can also work over a distance. And this particularly includes hormones like adrenaline, angiotensin, glucagon, gonadotropins. So they're released, enter the bloodstream, and then are transmitted throughout the body. So in addition to their importance in mediating responses to hormones and neurotransmitters, they mediate senses of sight, smell, and some taste. There are over 800 members of this family, and here's the family tree. As you can see, roughly half of these GPCRs are dedicated to the sense of smell. And this is a relatively small number for humans. For animals that rely more on the sense of smell, either to find food or avoid being food, you can see they have mice have roughly 1,000 odor receptors. Rats have 12,000 or 1,200. The largest number of odor receptors are found in elephants, which might make sense because they have the biggest nose. So there are a number of subfamilies. The largest family, we call family A, is the family that I've spent most of my career working on. They include adrenergic receptors, receptors for adrenaline, dopamine, for acetylcholine, dopamine, serotonin. More recently in my lab, we've been focused on opiates and cannabinoids, particularly because we would like to understand opiate signaling better and see if we can develop safer, more effective opiates. There's also family B receptors, and these are the target for a number of very popular drugs these days, the GLP-1 receptor, where agonists are used for weight loss. And then finally, family C receptors are important in neurotransmitter release and responses to neurotransmitters. So since they're the largest family of receptors for hormones and neurotransmitters, it's not surprising that they're the largest class of pharmaceutical targets. So roughly a third of all drugs on the market target G protein coupled receptors. And I don't have time to discuss this in detail, but given the very large number of receptors that we have, this is even a bit disappointing. And there are many challenges yet to develop safe and effective drugs for G protein coupled receptors, and that's an area that we're working on in my lab. So this is more or less the field in a nutshell. And now I'd like to give you sort of a brief history, because it's a relatively young field. And I would say it was born in 1971 when a scientist named Martin Rodbell identified that signaling through certain receptors required guanine nucleotides. And he was the first to propose that G protein coupled receptors exist. It wasn't until 1977 when G proteins were actually demonstrated biochemically by Elliot Ross and Al Gilman. And Gilman and Rodbell would ultimately win a Nobel Prize for discovery of G proteins. It wasn't until 1984 that the first G protein coupled receptor, hormone activated G protein coupled receptor, was purified. And this was done in the Lefkowitz lab by one of my colleagues. And as a result of that success, it was possible to obtain tools that ultimately would allow us to identify the gene for this receptor. So I joined the lab at Lefkowitz lab in 1984. Very fortunate time to join the lab because the protein had just been purified. And then we could use information from that purification to obtain a genomic clone for the beta-2 adrenergic receptor. So again, this is a receptor for adrenaline. It's a receptor that's particularly important for treating lung diseases such as asthma and COPD. And from the genomic sequence, we could deduce the amino acid sequence, and we could find that there were 7 stretches of very, what we call hydrophobic, fatty-loving amino acids. And that gave us the clue that this was probably a receptor that didn't just pass through the membrane once, but it actually passed through the membrane 7 times. And this has become a signature of G protein coupled receptors, this 7 transmembrane domain. Now, this is not a 3-dimensional structure, it's more or less a 2-dimensional structure, but it was the first clue that we had as to how these worked. Now, this is a very big discovery in the field, and particularly in Bob's lab. And so this is my mentor. I know he looks a little different here, but he was so excited, he went out and bought a Porsche and put on what we call a vanity plate with Turbo Bob. So the question was now, can we ultimately learn what these look like in 3 dimensions? And this is where I first had the idea that we could obtain structure using methods that were relatively new in the field of chemistry, protein crystallography. And it was the first time that I really thought about doing this. But as you'll see, it took quite a while before we succeeded. So again, this is cloning of the beta receptor in 1986. We also cloned a number of the other receptors, beta-1 receptor and alpha-2 receptor. So these are also adrenergic receptors. And having more than one allowed us to do experiments where we generated what we called chimeric receptors. And this is a project that I did with my wife, Tang Sun, who was helping me in the lab. And what we did is we took 2 receptors that had quite different properties. They both bound adrenaline, but they had different other drug preferences and different G protein preferences. And so we made a bunch of what we call chimeras with partial, some of them had a few domains from one receptor and from the other receptor. And so we had this series of chimeras, and we were able to, by studying the behavior of these chimeras, determine that this loop down here was important for G protein coupling, and that several of these transmembranes were critical for agonist binding. So it was our first clue as to sort of the functional map of these receptors. So in 1990, as I mentioned earlier, I started my own research program at Stanford. It wasn't until 1995 that the structure of a G protein was actually determined by crystallography, and not by me. This was by, again, Ross and Gilman. In 2003 is another important year when the human genome was sequenced, and it was then that it was determined that there were 800 members of this family. I would say when I joined the Lefkowitz lab back in '84, we might have thought there were 10 G protein coupled receptors. We had no idea it was such a big family. But it wasn't until 2007 that the structure, the high resolution structure of the beta-2 receptor was determined in my lab. In 2011, the structure of an active receptor was determined, again in my lab. And then finally, in 2011, the structure of a receptor G protein complex. And I believe it was this particular project that earned me a Nobel Prize. Now, if you look, I started in 1990 with the idea of getting a structure of these receptors, and it took roughly 17 years before I succeeded. So why did it take so long? So I hope to introduce you to a bit of technical expertise here in terms of protein structure. So how do you get the structure of a protein? So we have a protein, we know it resides in the membrane. We have to purify it in some way, and then try to coax it to form crystals, like your salt crystals or sugar crystals. Proteins can also form crystals. If you then take that crystal and bring it to an X-ray beamline and shine X-rays through the crystal, and in several different orientations, the diffraction patterns as X-rays bounce off the atoms of the protein can then be used to calculate a 3-dimensional structure. So this was my goal, and I thought it wouldn't take that long. So the first challenge was to be able to make the receptor in large quantities in purified form. And so we made modified beta-2 receptors. We were expressing them in insect cells, which happened to be the cheapest way of expressing large amounts of protein. We went through a series of purification steps, and then this is what we call an acrylamide gel, and you can see that at the very end, we had one band and really no other proteins that we could detect. So we had quite a good system for making protein. We could then use this for biochemistry, for spectroscopy studies, and the real goal was crystallography. So we were quite excited, but spent several years with this protein, and we were not successful. And it took until about early 2000 that we we identified one of the major problems. I'm sorry, my nose is running and I have no tissue. At the time we were doing these early crystallography studies, we thought of the protein as being fairly rigid. In fact, we had a two-state model. I know there are 7 transmembranes, but just to illustrate this, we're just using 2. And that the beta receptor without any drug or an antagonist was in a closed state. When bound to an agonist, it would open up, and that would allow G proteins to engage the receptor. But because we were doing other kinds of studies, particularly spectroscopy studies and studies that look at the sort of dynamic character of the protein, we soon realized that this model didn't work. And in fact, the receptor behaved quite differently. And again, these are using different methods besides crystallography, fluorescence, EPR, and NMR spectroscopy. And what we found is that the beta receptor is highly dynamic. That is, these transmembrane domains aren't in one position, but they oscillate and back and forth. And when you add an agonist, you can increase the probability that the receptor will be in an active state, but most of it will still be inactive. And only when you have a protein, such as a G protein, will you stabilize the receptor in a fully active conformation. So this is what we call a molecular dynamic simulation, and it tells you something about the real character of these receptors. So they're conformationally heterogeneous, and agonists alone won't stabilize the receptor in an active conformation. So why does this matter? Well, if you can imagine the receptor is really well behaved in a single conformation, then you can represent it by these hexagons. And these hexagons will pack together nicely and form a high-quality crystal. However, if the receptor is dynamic, it's conformationally heterogeneous. In other words, we have purified protein, but they're in different shapes. And as a result, they don't form a nice crystal. So in order to overcome these, and these are sort of the biggest challenges to getting the structure, one was what we call protein dynamics, this flexibility. And the other was that much of the receptor was embedded in a lipid bilayer or a surrogate for the lipid bilayer. And so we took 2 approaches. We developed antibodies that would stabilize the protein, or we used protein engineering. We modified the receptor with a more crystallizable protein. And it was in 2006, 2007 that this began to work. And we had our first crystals. So this looks like a nice high quality crystal. And you can see when we bring it to the synchrotron, we now get what we call diffraction patterns. And these dots are X-rays bouncing off the atoms in the crystal. And so we had the antibody complex worked. Also, the modified protein worked, again, using a special kind of media to grow the crystals. But from these, both we were able to obtain crystal structures of the beta-2 adrenergic receptor. So this is our first view of what it looked like in 3 dimensions. So the next step was to try to get an active state. So we have the inactive state, we want an active state. We, again, use the same tricks with making what we call an engineered protein bound to an agonist. Unfortunately, it crystallized in an inactive state, even though it was bound to an activating ligand. And again, remember, the protein is dynamic. Agonists might increase the chance of being in active conformation, but most of it's still inactive and get crystallized in that way. And we needed some sort of stabilizing protein. At the time, we were working on G proteins, but that wasn't successful. And so we identified a source of antibodies, and that was llamas. So llamas have special kinds of antibodies that are called single chain. So antibodies that you and I have heavy and light chains. Llamas have single heavy chains. And as a result, the variable component is oval-shaped and very good at stabilizing proteins. So we were fortunate to be able to obtain nanobodies, and in fact, those nanobodies stabilize the receptor in an active conformation. So this is the first view of what an active state receptor looked like. And, but at the same time, we really wanted to understand how a receptor activated a G protein. And this was a project that had been failing until we engaged a number of really talented collaborators and also postdocs and students in the lab. It involved expertise in biochemistry of G proteins. We had to find a very high affinity agonist by screening over 60 compounds from different companies, special detergents. Again, nanobodies were involved. We used protein engineering. The project was guided by low resolution methods, such as a single particle EM, and then special lipids. So as you can see, we succeeded, and I'll show you in a minute, we succeeded, but largely because we were very fortunate to have so many collaborators who contributed to this project. And this is the group that mainly were mainly focused on this project in the lab. My colleague, Roger Sunohara, his graduate student, Brian DeVry, and myself, Andy Cruz, and Soren Rasmussen from my lab. And these were our first crystals of the receptor G protein complex. And this is a very exciting time that we were just coming back from the synchrotron and took this picture. So we now have sort of a complete story, largely, and this was the receptor G protein team, most of them anyway, celebrating in Hawaii after getting this story published. So I'll just briefly show you what we learned about how this receptor works. And I'm going to go back and forth between inactive and active states. Okay, this isn't working, but so in the inactive state, you can see the agonist adrenaline is binding up here. The G protein hasn't engaged yet. And to activate the receptor, this transmembrane moves out. It allows this helix of the G protein to engage, and then this initiates conformational changes throughout the G protein that lead to binding to GTP. There we go, back and forth between active and inactive states. So at the time we had one receptor G protein complex structure, and we wanted to know whether what we learned applied to other receptor G protein complexes. So there are 4 families of G proteins, and there are, as you know, many different receptors that couple to different G proteins. We had many complexes that purified very nicely. We could not get crystals. But this was around 2017. X-ray crystallography was now being overrun by another technology that didn't require crystals, and that was cryo-electron microscopy, where you can simply take many pictures of single particles, single protein molecules, and then from them, using special detectors, special algorithms, and then refinement techniques, it was now possible to obtain a very large panel of G protein coupled receptors. And what we learned is that receptors that couple to different G proteins more or less worked in the same way. The receptor conformational changes were the same, and the G protein conformational changes were the same. So just to summarize, these are insights from these receptor G protein complexes. So all of these complexes are what we call nucleotide free. So we're capturing the receptor G protein complex after GDP is released and GTP binds. And that's the only way that we can get a stable complex. The structural changes in the different G protein families are very similar. The alpha helical domain of the G protein shown here is very flexible and very dynamic. And again, it's probably this dynamic character that made it difficult to get crystals. And unfortunately, these receptor G-protein complexes don't fully explain why a particular receptor will couple to a particular G-protein. So why does the beta-2 like the G-protein GS? Why do opiate receptors like G-proteins GI? And we think that specificity may be determined by a state that we really don't know for sure exists, but we think there are intermediate states that are very short-lived, very unstable, that are critical for this specificity determination. And this is an area that we're working on right now. In the last few minutes, I'm just going to mention a little bit about, okay, why do we care about these structures? Well, these structures can be used to help develop drugs. And the simplest way that I can explain this is, it's just one method, is it can be used to computationally identify drugs. So most drug discovery conventional drug discovery uses libraries of chemical compounds that are relatively small. It's 1 million sounds large, but you'll see it's quite small. Limited diversity. I mean, a lot of these molecules look like each other. You have to experimentally test each compound on cells expressing receptors, and the process is very slow and expensive. However, structure-based drug discovery allows you using docking algorithms together with crystal structures. The compound libraries are virtual libraries. They're very large, can be up to 500 million, high scaffold diversity, and it can be faster and much less expensive. So the computer basically takes each compound, it orients it in multiple different ways and sees if it fits into the crystal structure. If it fits, it gets collected. If it doesn't fit, it gets discarded, and you have a rank order. And just briefly, we were able to use this. Our first effort to use this was with the opiate receptor, where we identified a compound by virtual screening. This was in collaboration with the Scheiket lab, who developed a lot of these tools. These are the top scoring hits in the binding pocket. And from these, we selected compound 7. And then we again could use the structure. So this is compound 7 in the structure of the receptor to help modify the protein. It can improve its affinity by about 1,000-fold. And then we determined that it actually has some functional impact. So here's a pain relief assay from a mouse. It's called a hot plate. So you can see it, morphine is very effective at relieving pain. Our compound PZM21 is also very effective, and it actually lasts longer. And what's most important, it lacked the most troublesome aspect of opiates, in that the reason people die after overdosing on opiates is they stop breathing. And our compound, unlike morphine, which, as you can see in this animal model, reduced the breathing rate quite a bit, our compound, shown in blue, had really no effect on breathing rate. So what it shows you is that it is possible to use structures to obtain useful potential therapeutic, and structures are also useful for optimizing therapeutics in a number of other ways. So I'll finish here. I'll say that I've been very fortunate throughout my career to have very talented students and postdocs. That, as I mentioned, the collaboration of my wife throughout my academic career, and a number of very amazing collaborators. And this is the lab the morning of the Nobel announcement. So I'd be happy to, I guess, take any questions or.
Thank you, Professor Kobilka. If you want to take your seat here in the middle, and I would like to invite on stage Gabriele Beccaria, a science journalist for La Stampa and Repubblica. Welcome to Trieste Next, please.
Thank you very much. So, good evening, everybody. Thank you for being here. Thank you, Professor Kobilka. Check this microphone. Okay. Thank Professor, I saw the audience paying a great deal of attention to your speech, and I would say that we had a speech of what scientific research is and also what the life of a scientist is. So while listening to you, many questions came to my mind. And so I would like to start from the beginning, or at least I would like to ask you how you remember the day when you lived through the eureka moment, when you thought, well, I made the discovery that I was pursuing for years and years. How did you live that special moment in your life?
It was a moment that I guess it happened. I was in China at a meeting, and my students and postdocs were collecting data. And so I remember constantly emailing back and forth until we finally determined that we got the structure. And it was just, it was very excited. Although, because I had seen the crystals and they look really nice, and somehow I had a gut feeling that this might have been it. But yeah, it was very special. It was a goal that I had from very early on in my career, and finally we achieved it.
And so now we are a Nobel Prize, and I think that we are all curious to know how a Nobel Prize is changing the life of a person and of a scientist. I mean, we suppose that it was like a tsunami of events and also invitations from universities and special events. So how it really, if changed your life or not, and maybe how it influenced also your following research.
So it's obviously it's a great thing to have happen to you as a scientist. It's also quite disruptive, as you mentioned. The number of emails that you get on a regular basis goes up. You're asked to write letters of recommendation for people, and just a number of opportunities present itself for travel. And we've really enjoyed being able to travel. We've traveled really all around the world, although there are a few places we haven't been yet, but it was a great opportunity. But it's been a bit more challenging to keep on top of research because of that. I was fortunate that at the time that I won the Nobel, I had a very mature lab, and they didn't need me that much. But the other thing that I think up until the Nobel, I was still fairly active at the bench myself. I would help people with their projects, particularly projects like this where it's a higher risk. So I would sort of be people's technician, and I more or less stopped doing that because of other obligations, and I miss that.
Okay, and in your speech, you mentioned many times the drugs and their functioning. So I think that many of us would like to know something more about new drugs. I mean, your discoveries, how can pave the way to a new generation of drugs, maybe more effective and also more personalized to each individual.
Well, I think that pharmaceutical companies are still probably searching for drugs the old-fashioned way, but it's now much more common and much more frequently used in silico screening. There are new methods also for developing drugs that we've started using called DNA-encoded chemical libraries. But I think one of the problems is that we are beginning to realize that receptors that are important targets, for example, the opiate receptor is an important target. And this receptor, it doesn't simply signal through one G protein, it signals through 6 different G proteins. as well as 2 non-G proteins called arrestins. And it's very likely that the beneficial effects of opiates, that is relief of pain, may be working through a subset of these G proteins, and that the adverse effects are working through others. So we have to rethink how we're developing drugs and make them more selective for specific signaling pathways. And I don't think the pharmaceutical industry is really doing that the way they probably will in the future.
So not yet.
Not yet,
We have to wait for the future. So since you talked a lot about research and your life in the lab, I would like to ask you, we all know how difficult and challenging is the process of making research in a lab. So in your experience, what has to be the correct attitude of a researcher in order to get his or her goals? I mean, we know that a researcher has to be ready also to face failures. It's not. I mean, you don't succeed every time, right? So research takes time. So how can you be so resilient and passionate about research in order to go on and on?
Yes, I would say I started the lab in 1990 with the idea of getting a structure, and but it wasn't until 1995 that we really had the ability to make enough protein. So from 95 to 2005 were pretty much trials and failures. We would set up crystallography trials. We'd fail. We'd get occasionally we would get crystals, but they weren't protein. They were salt or lipid or detergent. And we would always, a major goal was to, when you set up an experiment, make sure that you can, at the end, if it fails, troubleshoot. So save everything during the experiment and try to learn something from failed experiments. And, you know, we had many failed experiments, so we learned a lot. And I think that the biggest challenge was to keep people excited and motivated about moving on when things weren't working well.
Okay, and so now I would like to talk about the present, and I mean, I would like to know, if you want, your comment on the present situation of scientific research in the US. I mean, we know how the Trump administration is changing the policies towards science, and we know that the situation is really worsening. So how worried are you about what's going on, about the cuts of federal funds and the shutting also of some agencies, or the threatened one?
Yeah, so I'm concerned on several levels. I guess more close to my own career, I'm not so much worried about my lab. I think we'll be okay. But I'm worried about my students and postdocs. You know, facing a more challenging job market, and also funding issues, which starting out a lab requires a lot of support from the NIH, and the current administration is just not supportive of any kind of science. And also, I guess the other area of concern is sort of how it might affect healthcare for the poor, and also just sort of the denial of the role of vaccines in health, and how that's probably already affecting children in a number of states where they're not being vaccinated against measles, and there are deaths now from childhood measles, which I don't think we've had for many years before that.
And so when you started your research at Duke and then at Stanford, it was easier to get funds than now, or the situation was very different, or not?
I would say it's definitely easier to get funds. I was very fortunate. I had funding from Howard Hughes Medical Institute to start with, and my first grant was funded. It was first R01 grant. So I know that prior to the current administration, the NIH grants were even the grant review process was designed such that young scientists would have a better chance. So their score didn't have to be quite as good as a more seasoned investigator to get funding. And I don't know what's happening to that. But yeah, it was. I would say grant funding has been becoming slightly more challenging up until the current administration, but now it's much worse.
And when we met earlier, you talked about your experience as a cardiologist and in an intensive care unit. So you worked as a physician for some time. So I would like to know how important was this experience with people in order then to move to a lab? What did you get from that very moment?
Yeah, I'm often asked this, and I don't really know that it has had a huge impact on what I do, but probably it definitely got me involved in this area of research. So I really wanted to know how these receptors worked because they were so important in terms of treating patients. But once I got into the research and discovered what they look like, and I really had to kind of change my mindset from physiology to biochemistry and structural biology. So yeah, it moved me in the right direction, but then I had to learn new things after that.
And so talking about the lab again, when we talk about scientists, sometimes we think of scientists as people, let's say, isolated people or working in a sort of ivory tower, but it's not like this. And you often said how important it is for a scientist to be connected with other people and scientists, and also how important are the human relations. And we know that also your wife supported you a lot from the beginning. So, and talking also about Professor Lefkowitz, I know that you became friends, right? So it was important, I think, this human aspect in your research.
Yes, I would say my mentor ran his lab in a sort of a different way than I run my lab because he had so many people. But he had the ability to, when things didn't work, he wouldn't, turn away and find someone else who was doing something that had succeeded. He would try to, boost your morale and convince you to, keep trying. And I remember when we were cloning and things weren't working so well, he would occasionally come and just sit in the lab and talk to us while we were working. And yeah, so he was a really a motivator. I had a sort of different approach. I like to actually be in the lab and working with people. But as a result, I couldn't mentor as many people as he did.
And so now you think that you are also a motivator with your students and postdocs?
I try to be, yes. Again, I've been very fortunate from the very beginning to having really talented people join the lab. And it's been a lot of fun to work with them.
So can you just tell us how important it is to work in a lab in a huge and important and rich university like Stanford? I mean, which is considered as a stronghold of research and teaching also. Why are you so lucky to be there?
Yeah, so why am I so lucky to be there? That's a good question. So I think one of the reasons that I was accepted is my department, molecular and cellular physiology, was a brand new department. And the chair of the department was a person named Richard Chen, who taught me physiology in medical school. He claims to have remembered me. And so I called him looking for a job. He was at the very end of filling positions. I think he was really tired of taking people out to dinner, interviews, and so I caught him at a good time, and I was able to land my first job. And it was a special department because we were all young, except Dick Chen, and I had pretty good financial support. Students that go to Stanford are pretty smart, and a lot of postdocs who applied, I think, didn't really even know what I did, but just wanted to come to Stanford. So it was a good place to start.
And you still have many foreign students coming to Stanford?
Yeah.
You didn't have problems with visas by now? Not yet?
Not yet.
Maybe in the future.
Yeah, I have one person who's making me a little nervous, but yeah.
And so, since I know that there are many students here in the audience, I would like to ask you if you can give us some suggestions to these young students if they want to undergo a scientific career, what they can do, what's the correct attitude in order to succeed in such a challenging field like research.
So I find this difficult to answer because I didn't get where I am because some sort of forethought or plan. I ended up being at the right place at the right time with the right people. And I think the one thing that might have helped me is I found something I was very interested in understanding. It was a big challenge because when I first started, we knew nothing about what they look like, and gradually over time, we solved small problems that ultimately allowed us to achieve our goal. So I was passionate about what this area of research was, and it kept me motivated. And I had particularly the support of my family and my wife when through good times and bad times.
Okay, so I would say as a final question, Professor, I would like to know, well, you are a Nobel Prize, but I suppose that you have still a dream, maybe, if not a secret dream, but a dream that you can share with us tonight about your research and future discoveries.
So I still have a real interest in how these receptors work. And the way I presented it, it might have sounded like, well, we know everything. But I mentioned at the very end that we still don't understand something very important, which is how receptors connect with specific proteins. And that's because there's a property of these receptors that is this dynamic character that I talked about. And I'm really interested in understanding how that directs the function of the receptor. So I still have a really important scientific goal. Probably is not, would put most people to sleep, but I find it very interesting. And the other is to really try to use what we've learned so far to be able to develop drugs that are more selective, safer, and more effective. And one of our goals is to develop better opiate receptors, or opiate agonists.
Okay. So thank you very much, Professor, for this lecture and your comments. And we hope to welcome you back again to Trieste in the near future. Thank you very much. Thank you all.
Vi ringraziamo per questa serata. Thanks, Professor Kobilka. Thanks, Gabriele Beccaria, for joining us tonight. e speriamo appunto che questa serata speciale di Trieste Next sia stato un momento di approfondimento e di scoperta della scienza, che poi è l'obiettivo di questa manifestazione. Vi ricordiamo nell'augurarvi buona serata che Trieste Next 2025 continua domani. Domani mattina appunto a partire dalle 10 avremo il premio Trieste Next, poi fino all'ora di cena per la giornata conclusiva di questo splendido Festival della ricerca scientifica. Grazie a tutte e tutti e buona serata. Grazie. Thanks again.